Genetic Analysis of Ozark Cave Bacteria, presented by Emerson and Thomas Maloney. I, Thomas Maloney, will be covering Introduction, Objectives, Collection, and Cultures, and my partner Emerson Smith will be covering DNA extraction and sequencing, results and discussion, and acknowledgments. Genetic diversity in cave ecosystems is largely unknown. There are two unique factors about Ozark caves. The first is that the constant temperature in the cave is about 12 to 14 degrees Celsius. The second part is that large regions in the cave are almost totally dark. We hypothesize that the microbes are still present and that they use light-independent sources to acquire energy. The environment is a good analog for potential life elsewhere in the solar system, and learning more about cave ecosystems will aid in this search. Our project had four primary objectives: to identify established and novel microbial species within caves using DNA barcodes, determine phylogenetic relationships with bacteria within and between caves, determine the total microbial biomass in cave soils and water, and monitor cave diversity over long periods of time. Through cave exploration, we were able to collect various cultures of bacteria. All exploration was done by NSS guidelines. We used sterile swabs and loupes to collect samples. Water droplets were collected as well when it was appropriate. We used various augers to collect our samples. BG11 auger was used for photoautotrophs. Boston's basal salt auger was used for chemoautotrophs. 1/2 Colobacter auger, 1/4 R2A auger, and hypho microbiome auger were used as well. Plates were incubated at 13 degrees Celsius for 1 to 12 months. BG11 was incubated under cool white fluorescent tubes. All others were incubated in darkness. Once pure cultures were obtained, they were streaked onto slants of the appropriate media. With coliform bacteria, Petri film was used. Samples were incubated for 18 to 24 hours at 37 degrees for total coliforms, or 44 degrees for fecal coliforms.
DNA extraction of the bacterial samples was performed with Wizard genomic DNA purification kits. PCR amplification was carried out using two different sets of primers, M13F27 Funif-backed and M13R1492R long-backed, which amplify a fragment of the 16S rRNA gene and was used to identify heterotrophic bacteria. P23SRVF1 and P23SRVR1, which amplifies a fragment of the 23S rDNA plastid, and was used to identify autotrophic bacteria and algae. The PCR amplicons were sequenced at GeneWiz to determine the nucleotide sequence that constitutes the DNA barcode. The two universal primers M13F and M13R27 were used for DNA sequencing. The basic local alignment search tool, or BLAST, available at the National Center for Biotechnology Information website, was used to identify sequences in databases. We identified many different species of bacteria, shown in the table to the right. Most bacteria identified so far belong to the phylum Proteobacteria, which is consistent with what has been described previously. Some bacteria were not identified. These could be novel species, or may not have been sequenced properly due to sample problems. Blanchard Springs Caverns, Cave Point Cave, and Cushman Blowing Cave had the largest numbers of identified microbes. However, most of this bias is because we have been working on these three caves for the longest time. DNA barcoding can be used successfully to identify bacterial species. The research is ongoing with the hope of identifying an even larger range of microbial diversity in the Ozark cave systems. Future studies will also involve phylogenetic reconstruction of the species identified. We want to give a special thanks to the members of the Cobra Grotto of the National Speleological Society for their assistance during the caving trips. This research is funded by the Arkansas Space Grant Consortium, Arkansas Embry, and Lyon College.